already running a minute late, but that's okay. We're gonna get going. We're gonna have some fun. Should be a good time. Got Dennis here with me. Hi, Dennis.
Morning, everybody.
So yeah, it's kind of a big topic, but we're gonna have some fun with it. We're gonna go into some detail that's probably excessive, but you know that's what I do sometimes. So today we're talking and going into some of the specifics of plant immune system and how the plant's immune system functions, which is a massive topic. And I mean, quite honestly, would take a whole lot more than the roughly half hour we're going to give it today. But it's a fun topic. There's a lot to it, and it's pretty interesting stuff. So here, this first picture. I love scanning electron micrographs. This is rust, wheat rust, and you can see all those little individual guys popping out, ready to move on to the next plant and get that infection going elsewhere. So the topics for today, we're going to start off kind of thinking about the world around you. What is an immune system? People versus animal, people, animal, and comparing that to the plant. So trying to make some similarities there so that we have a little bit of a stepping stone to bridge between the idea of a human immune system or a person or an animal immune system and a plant immune system. And we're going to zoom in, hopefully not too far, onto the plant immune system, and then kind of zoom back out. And then while we're doing that, we're going to talk about systemic acquired resistance and induced systemic resistance a little bit in the plant. So as I started working on this, it fairly quickly became apparent that this is a massive topic. There's a lot of information. And I like this quote by William DeMille. I've always admired the ability to bite off more than one can chew and then chew it. So that's what we're going to work on today. We're going to take off some bites. We're going to try to chew them together, knowing that this is a really big topic. It's a fun topic, but there is a lot there. So starting off the idea of the world around you, what is your favorite place in nature? And I've used this slide before, but it's a fun one, mental exercise. Imagine yourself in that place in nature. The sights, the sounds, the smells, all the things going on around you. Okay, so now imagine that you cannot move or speak. Basically, in other words, imagine yourself as a plant. Now that you can't move or speak or basically do many other things that we're used to being able to do in that environment, what struggles would you face? Dr. Ian Baldwin of the Max Planck Institute says the plant's genome is probably an order of magnitude more genes involved in environmental perception than most animals do. Most plants have to. They have to really tune their physiology and biochemistry to what is going on, and they need a very sophisticated system of perception and response. And it's kind of that idea of perception that is so critical. Perception, in this case, is primarily just the ability to access information from the outside world. What sort of information can be gleaned from what's going on around you? So here, the picture on the left, this is inside of your nose. This is an olfactory ganglion. Or basically a scent receptor. So what you got there is a structure with what kind of looks like a hand with little fingers dangling down into the environment. And that's basically, those have all sorts of little tiny receptors on the surface of those little quote unquote fingers that are going to capture scent molecules out of the air and then translate that information into our bodies, into our brain. The picture on the right is kind of the same sort of idea, but this is the surface of a plant. A plant cell membrane, and what we're looking at is a fairly similar finger-like structure, and again, the main function of that is to receive molecular information from the outside world so the plant can have an idea of what's going on around it. A kind of interesting phenomenon, an interesting example of that. Dr. Mesher from Penn State was working with goldenrod, and what he saw was as the male fruit flies started to excrete pheromones to attract the females, the goldenrod on the surface of its cells, on the surface of its tissue, has kind of those exact structures we were just talking about. It has receptors that allow the goldenrod to pick up on those male fruit fly hormones and react to it. As it reacts to it, what it's going to do is it's going to increase insecticidal chemicals, those phytoalexins or plant secondary metabolites, and in doing so, it decreases the ability of the females to lay their eggs and damage the plant, which is a pretty incredibly sophisticated ability of the plant to, just using a few of those hormone molecules, the pheromones from the fruit fly, interact with the world around it, and then prepare itself for the world around it. But that's the kind of basic idea. Everything is energy. Somebody has to live off of somebody. And here you can see kind of a lazy frog hitching a ride on a snail. But that's the energy balance. Everybody has to live off of somebody. And for the poor plants, they're pretty much at the bottom of that food pyramid. They are the energy producers. So they are targeted by lots of different organisms. The picture at the top left, there's corn. It's looking pretty sick. It got a cold, quote unquote. It has some sort of pathogen going after it. And we can also see a caterpillar there that's eating the plant leaf. It's getting its food and nutrition off of it. So as I started searching around and looking for, well, what are some of the ways that these organisms and these plants defend themselves, I came across a pretty fun one, the gimpy gimpy, and I wouldn't be too surprised if you haven't heard of it. The gimpy gimpy is best known in Australia. And it's not a very affectionate name, but its other name it's commonly known as the suicide plant. So when I showed this picture to Dennis, he said, well, it kind of looks like an African violet leaf. Yeah, it's soft and fuzzy. You look at that, oh, I kind of want to pet it and touch it. You don't want to pet and touch this one. So what's going on there, rather than a soft trichome on the outside, this plant produces, they're actually silica, glass, basically hollow tubes that are covering the fruits, the leaf, and the stem. Basically, every part of the plant, except for the root, is covered with these little tiny hypodermic needles that are hollow. So if you brush against the plant, those are going to cause instantaneous pain just because they're being poked by hundreds or thousands of these little sharp, jagged needles. But inside of those little hollow tubes, the plant has filled those little needles with one of the most toxic substances known to the plant world, moroidin, and that's a molecule that is extremely stable. And once those little needles are in you and the moroidin starts being pumped into your body, it's been described as feeling like a combination of being set on fire, electrocuted, and burned all at the same time, and have battery acid or an acid dropped on your skin, all the same place, all the same time. And this goes on, it depends on the person, their systems react to it slightly differently, but this can go on for weeks and in some cases, months. And this is an extremely stable molecule. There have been examples of researchers that have worked with museum samples that were collected over 100 years ago. They touch it, they get spiked, and they have the same reaction. And this is something that if you don't get those little needles out and say you take a shower, there's a story of a guy who went to Europe and he wasn't used to a cold environment. This is about a decade later. That cold environment caused his skin to do what skin normally does when it gets cold. He had goosebumps, but that squeezed the skin, which squeezed more of that moroidin back into his body, and he had a secondary reaction roughly a decade later. This is nature's way of kind of defending itself. It's the outside world that it has to protect itself from, because there are all sorts of invaders, organisms trying to eat you, trying to get, basically it's all that energy balance, trying to get that energy from you, from some way. So this picture here is Bubble Boy. This is an immune disorder where basically you don't have an immune system. You don't have a way to protect yourself from the outside world. So you have to have a completely sterile system. So the immune system is basically the collection of cells, proteins, and structures that protect the living organism from infections, organisms, and invaders from basically the outside world. Everybody's trying to get some of your energy. So this is the body's way of defending itself and protecting itself from it. So if we start by looking at the human immune system, which most people have a general idea, and I have a nutshell up there because we're going to greatly reduce the amount of information here, because this is a topic that by itself would take a long time. I mean, like doctors go to school for what, 6 years, something like that, to help figure out how the body works and a good part of that immune system. So we're going to boil it way down. We've got two basic components, the innate and the adaptive immunity. The adaptive immunity is what most people think of when they think of our immune system. Well, I got sick when I was a kid. I got chicken pox. I know I can never get chicken pox again. Or I went to the doctor and I got vaccinated for MMR, or, you know, at least most people did. There's so many outbreaks going on right now that it seems surprising that anybody got some of those injections. But what's going on there is that's their body's specific immunity, the adaptive immunity. And we have specialized cells, our T cells, B cells, and there are special forms of those called the T and B memory cells. And they basically hold on to that information. They're like a library. They hold on to the molecular structures of those different diseases. And so our body has that in its library ready to go. And there are estimates that our T and B memory cells can hold 10 to the 14th different types of antibodies. So our body has an incredibly large library to hold on to that information. That's our specific immunity. And that's a type of immunity that a plant really doesn't have. It can't be attacked by Pseudomonas syringae today, learn about it, figure it out, store that information, and be ready for it tomorrow. If syringae attacks today, the plant heals itself, and then the syringae attacks again tomorrow. It has to completely relearn how to defend itself from that. And that's kind of the other side, the innate immunity. And our bodies have the same. And it's basically a non-specific immunity. And it's the body's ability to attack basically anything it defines and views as not self. And that's a big part of the immune system, is identifying self versus not self. And the plant has a similar ability. And then we go to the part that pretty much everybody views and thinks about most easily, and that's the anatomical or physiological barriers. And this is typically referred to as the first line of defense. So for a human, we've got our skin, and pretty much the same side of that, a skin for a plant is known as its waxy cuticle. And it's basically forming the same, it's doing the same function. It's creating a physical barrier to stop things from the outside getting to the inside. That's the easy one. That's the physical one. We can look at it. I can go look at my skin. I can look at a plant. I can see those physical barriers really easy. But then it starts to get a whole lot more difficult when we zoom in, because to see what's going on the plant and in those cells, we have to zoom in. And this is on a cell-by-cell basis. So every individual cell in the plant has some of these pathways. Excuse me. So we're going to have a whole lot of detail here. We're not going to focus on too much of it, but I do want to draw a little bit of attention. The top right, there's MAMP, recognition, and we can see a couple different things there. Flagellin, chitin, those may sound familiar to you. Many bacteria have a flagella, and that flagella, which is basically the tail or whip that allows that organism to zoom around and move, it is made partially of flagellin. So these cells have the ability to recognize those bacterial tails. Chitin is what makes up most fungi and insects. So having a chitin receptor allows the plant to recognize that. Then unfortunately we get into, and we have to dig into a little bit of vocabulary, because this is a small sampling of the reality. But these are some of the things that I kind of wanted to drill down and focus a little bit on today. So starting up here, gene for gene hypothesis. This was developed in the 50s by Harold Henry Flor, and it's basically the idea that there is a resistance developed by the plants on a gene for gene basis. The pest produces a molecule the host, the plants, has the ability to recognize. And inside of that gene for gene, there are two basic structures. There's the R gene, also known as the resistance gene. And this is the gene in the plant that produces a recognition protein. Basically, we talked about, like the goldenrod, there's that surface structure. It has the ability to recognize the pheromone. That's what we're talking about here. Most of these are, we can think about them kind of like a lock and key. It's a shape that fits into this protein, and that's how the plant recognizes it. The other side of that is the avirulence gene, AVR genes, and those are genes the pathogen produce, genes in the pathogen that produce a plant recognizable protein structure. Again, key and lock sort of idea. Moving a little bit past that, we have our PAMP and MAMP, pathogen-associated molecular patterns and microbial-associated molecular patterns. And that's basically just the structure. Molecular pattern, what we're talking about there, is just primarily shape. And then a little bit deeper into that, we have our PTI, which is PAMP-triggered immunity. And then we have our ETI, effector-triggered immunity. And effectors are just proteins produced by the pathogens to help with the invasion, and primarily produced to suppress the immune system or help the other, or otherwise help the pathogen survive. Then we have HR, which we'll touch a little bit on. That's the hypersensitivity response, or PCD, which stands for programmed cell death, which can happen in the plant. So the gene for gene theory of Dr. Floor, basically what we're looking at is the idea of the plant can either have resistance or it can't. If the plant has a receptor, like those goldenrod did for the fruit fly pheromones, it can recognize that structure and then it can respond. Another plant may not have that structure, so it can no longer respond. And a previous presentation I talked about for corn, we've actually lost, the plants have lost the ability, we've genetically bred out the ability of those plants to recognize some insect attacks. And what they'll sometimes do is they'll call in parasitic wasps. They've lost that ability. So we can breed that sort of information out. So if the plant has the receptor, if it has the lock, and the pathogen has the key, then it gives the plant the ability to recognize it almost immediately and then become resistant to it. The PTI digs a little bit deeper into that gene for gene idea. And again, this is the pathogen associated molecular marker pattern triggered immunity. So we have here some elicitor. An elicitor is just something that causes a reaction, elicits a response. Bind to the cell surface receptors, resulting in general immunity. So one way that I like to think about this is our skin, the plant cell wall, that's like the structure of a house. We've got the exterior walls. That's the main idea, the main barrier to keep things out. When we go into PAMPs, what we're looking at is this the surface of the cell. So my thinking there is this is kind of like if I have security cameras with facial recognition software. So again, with the gene for gene, the facial recognition software is what's allowing that plant to kind of instantaneously recognize, oh, I know what that is. I see that it has a flagella. I see that it has chitin. I automatically know that is present, and therefore I can respond. When certain pathogenic enzymes, step 2, attack the cell wall, the breakdown products are recognized as elicitors by the membrane receptors. So as the organism attempts to attack, it starts breaking down the cell wall, and the plant can secondarily recognize those breakdown byproducts and can react to those as well. So it can react directly to some of the structures, the molecular patterns, or the breakdown products as that organism is trying to attack. Step 3, some of the elicitors bind to cytoplasmic receptors, resulting in specific immunity, kind of like we've been talking about. Step 4, signaling molecules trigger cellular responses, including the production of defensive molecules. And this is where the plant starts producing pathogenesis-related proteins. It starts shifting its hormone response, and it's going to produce more phytoalexins, and that's part of step 5. So those phytoalexins are plant secondary metabolites, essential oils. There's a wide variety of different terms that are associated with those phytoalexins or protective molecules. Those are going to go after the pathogen directly. And some of those PR proteins serve as an alarm signal. So as that cell is being attacked, it has the ability to send out specific molecules or hormones telling the cells around it, as well as the plant as a whole, that it's under attack. At the same time, it kind of lets some of those molecules through, and then it'll shut the door behind it. It will secrete additional sugars, polysaccharides, extensins, to strengthen the cell wall and to stop up that plasmodesmata. And the idea there is, oh, this specific cell is under attack. I'm going to go after all the different organisms, whatever's going on, with my protective structures. At the same time, I'm going to block off that cell so it can't escape. So that's all the PAMP. That's the PTI. That's the outside of the cell. There are other things that can go on, and when they go directly inside the cell, that's the effector-triggered immunity. So the TTSS, that's the type 3 secretion system, which is basically just a fancy kind of syringe that bacteria use to bypass the cell membrane. They'll produce kind of a little pilus, a little syringe-like structure, and they can directly inject different molecules and proteins into the cell. Nematodes with their stylet, aphids with their stylet can do similar things. Fungus with a hostorium or mycelium, they can poke through, and basically they're just trying to get through that cell wall so they can start dropping in bombs. They can start dropping in things that are going to cause a negative impact or effectors, the immune system of the plant, and try to stop up the plant's ability to stop it. So the PTI is the security, the facial recognition. The ETI, these are the security guards inside of the cell. So there are specific molecules and structures inside of the cell that are designed to recognize those effectors. They have, the plant will produce a wide variety of these different structures. And again, it's like a lock and key. Inside of the cell, there are these molecules that recognize the effector. They're then going to go back into the genes of the cell, the DNA of the plants, and cause a massive production of protective molecules.
You know, and Steve, here's one of the places that I like to talk about. We stress so much about balanced nutrition, calcium, calcium needed for cell wall development. And when we start to relate that to how, you know, the PTI or the ETI affects the plant, a lot of that is directly related specifically to cell wall development.
Oh, absolutely. So I mentioned up here, these extensins, these cell wall strengtheners, that's part of it. And we could go into, there are several different structures and several different reactions that happen. There's also callose deposition, and there's reactive oxygen species that are associated with the callose production specific. There's, again, we're trying to take a small bite, but there is a whole lot more going on. But you're absolutely right. When the plant is attacked like this, it has to have the energy to be able to produce these molecules, as well as specifically for like calcium pectate to strengthen the cell wall. It has to have a lot of calcium to help close off and protect itself. So we talked about the individual cells, and we started to talk a little bit about some of those proteins and signaling molecules that the cell will produce as it is individually attacked, that's going to go out to neighboring cells, as well as through the plant's vascular system to warn the rest of the plant. We're not going to focus very much on abscisic acid, ABA. That is typically associated more with abiotic stress, so things like drought stress, heat stress, those are non-living stresses. Today we're mostly focusing on living stresses. So the SA, salicylic acid pathways and phytohormone, is mostly associated with pathogens and pathogen attack. And the jasmonic acid pathways are mostly associated with herbivory and insect attack. And I say mostly because it's a fairly complex system. This picture on the right, there's a whole lot going on with negative interactions, positive interactions, mobility, which phytohormones are interacting with each other, whether it's going to cause more plant growth, more new systemic resistance. We're going to make the bite smaller and focus more on just kind of the broad brush strokes. And that's basically the jasmonic acid is considered the main hormone regulating the switch between growth to defense through positive and negative crosstalk with other plant hormones. It's going to be basically dictating how well that plant is growing. When the plant is under attack by an insect, it's going to switch, it's going to massively increase its jasmonic acid, and it's going to massively decrease its ability to grow. On the other side of that, if we can increase our auxins, cytokinins, gibberellic acids, brasinosteroids, that's going to mostly be related to the plant's ability to grow. So if the plant is healthy, not being attacked, more growth. Once it is attacked and it switches its hormones, it's going to decrease its growth and switch that, again, that energy balance. It's going to switch that energy over to protection. So for our jasmonic acid pathways, step one, generally, insect wounds or plants that insect wounds plant tissue. Step two, the wound activates jasmonic acid. Jasmonic acid then flows through the cells, interacts with the proteins or DNA to activate genes. The enzymes and other proteins that defend against the herbivores are produced, and then they're also going to be spread throughout the plant. That's the basics of the jasmonic acid pathway. It's there to help protect primarily against insect attack. The other side of that, most of what we're talking about with the pathogen attack, the PTI and ETI, is more associated with pathogen and the salicylic acid pathways. So what we can see here, if we look at the plant picture kind of on the slightly upper right, we see step one, there's a specific infected leaf. So what's going to happen, all that PTI and ETI that we already talked about inside the individual cells is going to start and trigger a response. As that response happens, the plant is protecting the cell walls, increasing their thickness, producing all those protective molecules, and at the same time, increasing its salicylic acid hormone. It's going to then spread that salicylic acid to neighboring cells of those that are being attacked, as well as through the phloem, xylem phloem of the tissue, the vascular system, so that the rest of the plant can basically get a heads up of there's something bad going on. And interestingly enough, the plant, so we can see here, bottom right leaf, this is specifically where our attack is going on. These are the attacked cells. And we can see the salicylic acid levels go up or spread throughout the plant. And then this is where we also see the PCD, programmed cell death, or HR, hypersensitive response. So if you've ever gone out, looked at a plant, a leaf, a fruit, and you've seen little spots where it looks like, is this overspray? Is this drift? What happened here? Sometimes that can be the plant's protective immune system, meaning that that plant had been attacked by something and it had the ability to defend itself and fight that off. And basically all it's trying to do is isolate that infection to a small cluster of cells. And in doing so, increasing its salicylic acid pathways, it's going to then help protect the rest of the plant from an attack of a similar organism. Interestingly enough, the salicylic acid can also be converted to methyl salicylate, which will translocate throughout the plant, but it also acts as a volatile molecule. So if I have a neighboring plant, and this is pretty well studied and known in tomatoes, for example, when a tomato is attacked, it will produce molecules that basically warn the neighboring plant. So if the plant on the left, or plant A, is attacked, all those responses are going to happen. Salicylic acid is produced, methyl salicylate is produced, and then it's going to volatilize and basically warn the neighboring plants. They're then going to be less likely to be attacked. In a similar thing, kind of like we talked again, that goldenrod, it's a similar sort of response where it's a systemic, it's throughout the entire plant to protect itself. So this one I'm going to click over to, and this is kind of an interesting new bit of research, and it's basically completely accidental. There are some researchers that were trying to work on gravitropism. How do plants grow away or towards gravity? That's called gravitropism. What they were trying to do is figure out a way, how can we monitor how these plants are going to grow? So they modified plants to basically glow when the presence of calcium is regulated. So we can see here, this is basically over a couple of minutes. What they did is they took glutamate and dripped it on the plant cell, and they found since then that when a plant is crushed, squeezed, damaged, bitten by an insect or caterpillar, these glutamate levels will spike, causing the calcium throughout the plant to increase as well. And you can see once that kicks into gear, it only takes about 10 to 15 seconds to travel throughout the entire plant. This is separate from the jasmonic acid. This is separate from the salicylic acid. This is something new. They don't fully understand it yet, but what they found is it's pretty incredibly fast as far as plant response goes. And interestingly enough, it uses glutamate and calcium. Our neurotransmitters, our brain uses glutamate and calcium to help translocate and shift those neurotransmitter signals throughout our body. Our heart beating is caused by calcium efflux, moving calcium in and out of cells. So this response in plants, although plants don't have a brain, don't have a central nervous system, this seems to be a fairly similar response to how our brains send electrical signals throughout the body. And this is basically thought of kind of similarly to a pain response. The researchers were also able to show just brushing against the plant, they could have a similar response where you could see basically wherever they tapped or touched the plant, you'd see this, this flush. So it's kind of a touch sort of response. And they're still trying to figure out all of what it means for the plant. All right, let's see if I can get back into the presentation. All right, so we've talked a little bit about up top. We've got pathogen infection we talked about with the salicylic acid. We talked about wounding, jasmonic acid. There's this other side, and what we have up here is this is non-pathogenic rhizobacteria. We usually talk about plant growth promoting rhizobacteria, so our beneficial organisms. And they can do what's called an induced systemic resistance. And as you can see kind of on the bottom left, I circled and highlighted a few things. And that's primarily just to point out that they don't know. They know that this is a strong response. Scientists know this is a beneficial response, but exactly how it works and why it works, they don't know. There's some big question marks there, some big holes there, unknown defensive compounds. What we can see and what we know is that what happens is the term induced systemic resistance, generic term for the induced state of resistance in plants triggered by biological or chemical inducers, which protects non-exposed plants parts against future attack by pathogenic microbes and herbivorous insects. So just by having in the soil environment the presence of these beneficials, and the INR7 up here in the top left of our photo inset, I believe that is a Bacillus pumilus. Just having these organisms around the roots helps protect the plant from both insects and pathogen responses. And they've taken it steps farther than that. They can take the volatiles, the gases that are coming off of these bacteria, and expose the plant roots and tissue to just the gases that these organisms are producing. And in some cases, they'll see not necessarily as strong of an ISR response, but they'll see an ISR response just from not even touching, not even growing with, just having those beneficial organisms in the same vicinity around that plant. And this has been done here. Is cucumber beetles, field cucumber. This is Bacillus pumilus. Pretty fantastic results in reducing the level of the cucumber beetle, basically performing better than our typical conventional insecticides in this case. Incidence of bacterial wilt and field cucumber treated with PGPR. And again, this is our INR7, which is the Bacillus pumilus. And again, we see it outperforming our more conventional approaches. This is kind of a fun one. This is fairly recent research. See if this little video will fire up and work. So what we're seeing here, University of Delaware. So we can see this is Pseudomonas syringae actually growing and on the surface of the tissue. Oh, there goes one. Oh, so what we can see, those kind of lip red-shaped structure, that's a stomata. Those syringae are now inside the plant. Treated with probiotics, in this case Bacillus subtilis, the plant has a faster reaction, a faster response time to closing those stomata, and now those Pseudomonas syringae cannot get inside of the cell. The pathogen has been kept out, and that's simply by having, again, having the presence of the Bacillus subtilis in the root zone. It causes a fast response for the plant.
You know, when I see something like this, Steve, when we talk how quickly diseases can spread, this is why it is so important that we have healthy plants and we have a disease-resistant soil, which a diversity of cover crops, microbial communities, building soil health, so that all of these players are on the field. So when this plant is actually stressed or under attack, it already has those there.
Yeah.
You know, rather than as we see how quickly they can get into the plant, now we're going with some of our control products that you talked about, but we've already had some infection into that plant. So if they're already there based on a healthy disease-resistant soil, as they talk about a lot, and one of the reasons why we talk so much about building soil health, we can see the benefit of how that might all work.
Oh, absolutely. So we're increasing the available energy of the plant by increasing the amount of fertility available to the plant, as well as through these induced systemic response effects, we're directly impacting that plant's ability to, even before the incidence of disease comes in, the plant is basically pre-programmed, pre-wired. This is kind of like the goldenrod, sensing that pheromone. Those insects aren't even there laying eggs yet. Those fruit flies aren't even present. These beneficial organisms down in the root zone, they've pre-programmed, pre-wired. It's like your smoke alarm going off, but instead, the fire department's already there. They're already responding. You didn't even know what was going on yet. It's pretty incredible. And we've talked in the past, and you were just talking a bit about the idea of recruitment. So our plants are producing massive amounts of root exudates, and they're doing that purposefully. And they're using those root exudates in a very purposeful manner. So this is double whammy. I've used this before. We're talking about corn here, benzoxazinoids. And benzoxazinoids act directly as powerful antibiotics against aphids and fungi, So that makes sense. The plant's going to produce it and put it up in the leaf tissue where it's being attacked. It will also send those benzoxazanoids down through the root. And what the researchers has found is that it actually recruits or draws in Pseudomonas putida. The Pseudomonas putida acts through ISR to help reduce the incidence of the aphids and the fungi, as well as increasing the protection for those roots. Knowing that the plant is under attack by aphids and fungi, the health of the plant is going to go down. It's going to need more defense. It's going to be more susceptible oftentimes to a fungal pathogen as well. So as those Pseudomonas putida are attracted in, it's helping protect the top parts of the plant as well as the root of the plant at the same time. And we've seen similar things with malic acid. Plants will spit out malic acid through their exudates to recruit in Bacillus subtilis. We already saw Bacillus subtilis with Pseudomonas syringae, and this is another bit of research with Pseudomonas syringae. The researchers basically put the Pseudomonas syringae on the leaves and in the root zone of the plants. The plant starts producing the malic acid to attract in the Bacillus subtilis, and the Bacillus subtilis then grows. We can see it growing around the roots, protecting the root and the root hairs. Interestingly enough, there are a number of organisms, Bacillus subtilis, Bacillus vortexii, and a few others that they know, and they'll do what's called swarming. They don't know how, but these organisms are able to basically work in concert. They work together. These are single-cell organisms that are able to communicate in some way to basically program their growth and their response so they can overwhelm and surround different organisms. And they've seen this happen with insects, and they've seen this happen with disease. It's a swarming response where massive numbers of these organisms grow specific shaped colonies to help surround, digest, and protect. So when we talk about PGPR, we're talking a little bit about ISR, but they do so many other benefits. Having them in the soil, we see phosphorus solubilization, hydrogen cyanide production, enzymes, and as well as organic acids, zinc solubilization, silica solubilization. They're doing a wide variety of jobs. And a big part of that is because these organisms, plants, and the PGPR that help protect them have been growing together for a very long time. And they have to, because they are a part of the plant's immune system. They are a part of the plant's ability to respond to the environment around it. And again, I've mentioned this before, but it all boils down to energy. I mentioned that plants are basically the main source of energy, so of course they're under attack on a regular basis. They're our primary producers. They're the way that all of our systems that we recognize and know on Earth, that we humans basically interact with on a daily basis, that's where the energy for all of the living systems comes from. The sun, through photosynthesis, produces sugar. And that's basically what it all boils down to. That is the basis of energy for this entire planet. So everybody is going directly after those plants because everybody wants to live, everybody wants to survive. And so it does become a direct energy balance. So with all that in mind, what can I do? And most of what we talked about today is what the plant has the capability of doing on its own. It can do it. I underline has the capability of doing. It doesn't mean that it necessarily will be able to. The plant needs our help, and a big part of that is the energy side. Bruce used to talk about, he would often say, disease and insect attack happens for a reason. Disease is nature's way of cleaning house and getting rid of the garbage. When I have a low energy environment, when I have plants that cannot survive and cannot reproduce effectively, nature wants to get rid of those. They either don't fit the environment or their genetics are not strong enough. So what Mother Nature does is purposefully sends in pest and disease to help clear house. So what can we do? Through our best management practices and timing, we can reduce some of the stresses. And we've talked before about the idea of the plant's genetic potential. You can think about it as a fully inflated balloon. It starts with however much energy and genetic expression potential that plant has as a seed going into ground. Every bit of stress, everything we do wrong, everything we do right, has an impact on how big that yield potential balloon remains at the end of the season. Am I focusing on my timing? Did I plant when it was too wet? Did I plant when it was too dry? Did I plant too deep, too shallow? Have I been focusing on balanced nutrition? The timing, the form of nutrition makes a huge difference. Every one of these is kind of like, when we do it wrong, a little pinhole in that balloon of total yield. Paying close attention to deficiencies. And Dennis has been talking a lot lately about the idea of excesses can sometimes be more damaging than deficiencies to the plant. Trace minerals are critical. We've been talking a lot about immune response. The immune system requires a wide variety of different trace minerals to fully function. Zinc, manganese, iron, all directly associated with some of those protected molecules and pathways. Silica, the same, as well as strengthening our cell walls and membranes and reducing herbivory. These are all critical to protection and response. And our beneficial microbes are the key to digestion for the plants, and therefore the key to optimizing the energy balance. Without that digestive system, our plants cannot produce the amount of nutrition and energy that they need to fully perform and to fully produce.
And that comes so key to what you were talking about on photosynthesis and how the plant basically produces the energy and is able to utilize that energy in order to grow, produce fruit, and also protect itself. You know, one of the things that we talk about a lot is a form of nutrition that we're using. And some of those forms of nutrition actually take energy from the plant in order to convert it to make it into a usable state. So when we start to really think about that, anything that we do that takes energy away from that plant can also make it more susceptible to attack. Kind of what you were talking about a little bit based on that balloon and letting just a little bit of air out of that balloon. We've just reduced our genetic potential. So that is why we are always stressing what form of nutrition are we using and how important is that? And again, making sure that we don't create any excesses so that we have balanced nutrition within that plant.
Oh, definitely. I mean, looking at Francis Chaboso's work, the level of nitrate. Nitrate has to be converted by the plant. Every enzyme associated with converting that nitrate into amino acid takes energy. If we can go directly to an amino form or a biological form, which many of our bacteria are doing, we're decreasing the amount of energy directly the plant has to do. You had those pictures here not too long ago of, I believe it was some citrus, that we were looking at the leaf size and how the leaf size was dramatically increased. Again, that is our solar array. That's our solar panel. And every bit of that is the potential energy that the plant can produce, as long as we're supplying the nutrition that it needs, when it needs it, how it needs it. So I think with that, Dennis, you want anything else? I think we're going to open it up to questions and say thank you, everybody. So there is a question about salicylic acid supplementation, better applied via drench, foliar, or both. I am generally very cautious when it comes to applying any sort of plant phytohormone. My general recommendation is approach is let's utilize biological systems that have already developed those molecules in a fairly balanced way. And one of the ways we can do that through a foliar or a drench is with something like seaweed. Seaweed has a little bit of a wide variety of plant phytohormones. I've seen lots of examples where things go wrong. A little bit of phytohormone can go a very long way. And if we overdo it, we can force the plants into a protective mode or protective state that we didn't really intend to, which can then decrease the amount of growth. More growth is going to be typically associated with a better yield. So if we force that plant into protection too far, it's going to produce excessive protective molecules, which again, cost energy. So my general approach is let's let the plant and living systems do the majority of the work. I like seaweeds for phytohormones, and many of our PGPR through the roots, as well as their phyllosphere bacteria that are going to be producing a variety of different phytohormones, letting those natural systems basically dictate how much of that phytohormone is produced at any given time is my general direction. Dennis, you want to add to that?
Nope. I always get nervous when we start to try and manipulate the plant based on what we think is best. So again, I always look at, again, when we talk about our foliar applications, we're talking about using some fish, some seaweed, some trace minerals.
Nutrition.
Nutrition.
We're more focused on the nutrition side of it than the specific hormone side. We're letting the plant kind of dictate itself.
Yeah. And
Sorry,
let nature run, and let nature run its course.
Yeah. So I am extremely cautious whenever I suggest applying any sort of phytohormone. So I don't know if I have a direct response for that. Again, drenching or foliating a little bit of seaweed on a regular basis, I don't have a problem with that. We encourage that. We love that. As far as direct applications of 4-chlorphenuron, gibberellic acids, you have to be very careful because you can overdo it very quickly. Let's see. Yeah, Micro 5000, yeah, is another great form because it contains a good amount of seaweed in there, so it's going to do some of that automatically. Let's see. Well, I guess that made it pretty easy. No questions. We had the one, which was a good one. It's just one that I'm pretty cautious with answering. And if that is it, Dennis, do you have anything you want to add?
No, I think that's great. You know, I kind of go back to that very first picture that you had, Steve.
The Gimpy?
No, the rust spores on the wheat. And it kind of gives you an idea of how quickly and these pathogens can spread and how many spores are there and why all of this is so important, because it can happen so quickly when we start to talk about when the conditions get right, how quickly that plant immune system has to increase in order to suppress all of these types of things. And so, you know, I mean, this gives us a pretty good idea. I mean, you look at the number of spores there from the rust, and that is something that plant has to be able to overcome in a relatively short period of time.
Yeah, I wish that there was a scale bar on here. But looking at the size of the trichomes on there, looking at the structures, looking at the stomata, that's not too much thicker than a fingernail clipping or a hair that we're talking about. That's one little individual, you know, pustule that's producing thousands of these rust spores. So when you look at a wheat that is expressing rust and you see, oh, it looks like a whole bunch of orange, when you zoom in, you see it's thousands of these individuals with thousands of these spores inside. It is incredible how fast those can be. Soy protein is an end source taken up directly as an amino acid. Yeah, we have a lot of people that are really liking the protein approach. You have to be a little bit careful with it because, I mean, it is nitrogen. You don't want to overdo it, but we have a lot of people that are using the soy protein. We really like soy protein. The fish, I love fish fertilizers, especially ocean caught, because you have oils, you have fats, you have a wide variety of things in there, including calcium. And we have a lot of people that are doing kind of a combination of the fishes with, if they're trying to boost up their nitrogen, the soy. Most of what you're seeing there is glycine, which is fine. It's a very simple amino acid. It's kind of the base amino acid that most other amino acids are going to be made from. So the plant's going to directly take it up. It's going to utilize it. And then it's going to modify it into all of the other proteins and amino acids it's going to use. But yeah, much more efficient to use a protein form than almost any other form of nitrogen.
You know, and sometimes I talk about when we look at the form of protein or nitrogen that we're using, a lot of times I look at, are we feeding the plant or are we feeding the soil? So often if we're feeding the plant, the soy protein nitrogens are great because generally they're going to be 14, 16% nitrogen, where our fish is going to be more around that 3, 4. But the fish is a much better microbe food. And it also has trace minerals. There's a lot of benefits from it. So I think kind of a combination is not that the fish isn't wonderful to feed the plant. It is. But as Steve talks about, when we look at our nitrogen levels within our plant, we need to choose, you know, what are we trying to do and make sure we utilize that tool properly.
Yeah. And utilizing, I mean, we had David on here this last month looking at sap analysis, looking at the tissue or the fluid analysis basically out of those tissues. And that gives us a really great tool and basically a compass of what direction we should be going in with some of this nutrition as time goes on. And it's a relatively inexpensive, relatively easy process, and it helps give us a great indication of where we're at. Okay. Well,
I think that's it.
yeah.
Thanks, everybody.
Awesome.
Good job, Steve.
Well, thank you, Dennis. I had fun. Hopefully everybody else did, and hopefully the bites weren't too big.
I'll be chewing for a while.
I know. Thanks, William DeMille. All right. Well, thank you again, everybody. Thanks for attending, and we'll talk to you here next month.
Thank you, Steve. Well done.
Thank you. Bye for now.
Bye-bye.